Hi guys, Miss Francis here. I'd like to discuss the Krebs cycle with you. In class, we watched an animation, then we annotated this image, we took some notes, and we watched another animation. If after this video you're still lost, you need to go back and revisit the animation, the McGraw-Hill, how the Krebs cycle works. You can just Google it, as well as the Stephanie Castle video, which you can also Google. So let's dive into the Krebs cycle. Now, the Krebs cycle is also known as the citric acid cycle. Why is it called the Krebs cycle? Because the scientist who discovered it last name was Krebs. Why is it called the citric acid cycle? Well, because the cycle starts with citrate, which is a 6-carbon compound that forms when OAA, a 4-carbon compound, combines with acetyl-CoA, our 2-carbon compound. So I'm going to go through the steps of the citric acid cycle. So let's start with citrate, also known as citric acid. Right, so this is also known as citric acid, hence the name citric acid cycle. So citrate loses the water, gains the water, forming isocitrate, which is still a 6-carbon compound. This 6-carbon compound gets oxidized. In the process of it getting oxidized, NAD is going to get reduced and form NADH. So now we have our first NADH. Isocitrate is also going to get decarboxylated, and that enables us to get our next compound being a 5-carbon compound. So this 5-carbon compound is also going to get decarboxylated, losing a carbon and giving us our second carbon dioxide. It's also going to get oxidized, enabling NAD to get reduced, giving us our second NADH. So far, if we are keeping a running tally, we've got 2 NADHs and 2 CO2s. Back down to our citric acid cycle. We are now at our first 4-carbon compound, succinyl-CoA. From succinyl-CoA, you're going to experience substrate level phosphorylation, and we are going to get 1 ATP. Succinyl-CoA is not decarboxylated, so our next compound is also a 4-carbon compound called succinate. Succinate is going to get oxidized, which enables FAD to be reduced, giving us FADH2. So now if we return back to our running tally, we've got 1 ATP, and now we've got 1 FADH2. Moving along, fumarate is still a 4-carbon compound. Water enters the picture. Malate is still a 4-carbon compound, which will be oxidized, thus enabling NAD to be reduced, giving us our third NADH. So returning back to our running tally, 3 NADH. And that leaves us off back at OAA, a 4-carbon compound, which then can join again with acetyl-CoA, our 2-carbon compound. Now remember, for each glucose, this cycle is going to spin twice. So in order to determine our totals, we need to multiply each one of these running tallies by 2. So if I multiply this by 2, I get 6 total NADH. If I multiply this by 2, I get 2 total CO2. 4 total CO2. If I multiply this by 2, I get 2 ATPs. And if I multiply this by 2, I end up with 2 FADHs. So now let's fill out our note packet by identifying what you really need to know about Krebs cycle. So here's the most important part of the most important information that you need to know about the Krebs cycle. 2, I know this is hard to see, but 2-acetyl-CoA are the initial reactants, where the cycle begins when a 2-carbon acetyl-CoA joins with the 4-carbon OAA to form that 6-carbon citrate, where that 6-carbon citrate is also known as citric acid, hence the name of the cycle being called the citric acid cycle. Now, we need to remember that there are 2 spins of the cycle per original glucose molecule, because we end up with 2 pyruvates for each glucose, and each one of those pyruvates has to go through this cycle. So what does the Krebs cycle give us? Well, with those 2 turns, I'm going to get 2 ATP via substrate level phosphorylation, 4 carbon dioxide molecules, 6 NADH molecules, and 2 FADH. Remember, those NADH and the FADH are our electron carriers. So what are our totals at this point? Well, we've got 4 ATP, because we had those 2 from glycolysis. We've got 6 CO2, because we had 2 from glycolysis. We've got 10 NADH, because we had 2 from glycolysis, 2 from the link reaction, and our total of FADH is just 2, because this is the first time we're seeing FADH. This entire cycle occurs in the mitochondrial matrix, just like the link reaction occurs in the mitochondrial matrix. If you are asked to label the mitochondria, you should be able to recognize that the intermembrane space is the space between the inner membrane and the outer membrane, that the cristae are the folds within the mitochondria that help increase surface area, and that the matrix is the space in between the cristae. This picture represents those places a little bit, those structures a little bit clearer. Here's what a picture of the mitochondria might look like if you were to draw it on your own, as well as the locations of where the link reaction and the Krebs cycle occur, right here in the mitochondria matrix. We are going to learn that the ETC and chemiosmosis are going to occur on the inner membrane. Again, here is another version of a hand drawing of the mitochondria, an ultrastructure of the mitochondria, showing all of the different structures, where you can see we are headed towards the inner membrane for ETC and chemiosmosis. So before we get to ETC and chemiosmosis, I'd like to continue our flow chart so that we're up to date with that. In our flow chart, we are right here. We're up to the Krebs cycle. The Krebs cycle is going to take in 6 water, which I don't think I mentioned that earlier. From the Krebs cycle, we are going to make 2 ATP, right? We're going to make 4 CO2. We're going to make 6 NADHs, and we're going to make 2 FADH2s. And that is the totals with 6, that's the totals when you have 2 turns. In order for a Krebs cycle to be considered fully done, you need to have 2 turns of that cycle, because remember, for each glucose, I'm going to get 2 pyruvates.